מישהו פה שכח מים רותחים. אין בעיה, רק... אוקיי, בשיעור הקודם, אתם זוכרים, אנחנו מדברים... סורי. Pardon. Last time we talked about, started talking about olfaction. This is the second basic session on olfaction, and I'm not sure we're going to make it all the way through. Anyway, next week there's one free day. I don't know what, the Yom Ha'atzmaut, I think, or Erev Yom HaZikaron. And so there's no class, I think. And then the advanced talk this year, we try to present the new members of the Elsc community. So Yoram Ben Shaul from Hadassah, which is an investigator here, will give you an advanced talk on the topic of olfaction. Okay, last time we left here, where you remember there are 1,000 roughly different receptors, odorant receptors, which send in the epithelium, sending their axons to these discrete structures called glomeruli, where all the axons converge. And in 2008, the group of Richard Axel made a mouse, which they call a monoclonal nose, and they forced all the neurons in the epithelium to express a single odorant receptor out of the 1,000, one for which there is a known ligand. It's not the best ligand for this odorant receptor, but nevertheless, it's a ligand that activates it. So basically, they made a monoclonal mouse, monoclonal nose mouse. Where supposedly, it can smell only the ligands that are activating the receptor, which is expressed in this animal. This is just some proof that indeed, so there is some residual genetic noise in the control in the animal, but basically, if you take this, the receptor is called M71. So the M71 transgenic, if you give a normal mouse, either, and the ligand is acetophenone, sorry. So the M71 is the receptor. Okay, it's the odorant receptor. And the ligand which activates it is called acetophenone. It's one of many. You remember from Linda Buck's paper, acetophenone. So this is one ligand. I told you that these are orphan normally. Most of them are orphan. We don't know which ligand activates them. For this receptor, we do know a ligand that activates it. So they made a mouse that the whole epithelium is expressing this receptor, and they know that its activation. And if they take a control mouse and you give a puff of acetophenone or a cocktail of odors, you will get this kind of activation. Okay, and it's a small blimp. These are recordings from the olfactory nerve, from the epithelium to the bulb. You see a small blimp of activation. One odor, which is the red, the acetophenone, will give you a small blimp. A few thousand axons will be activated. If you give a cocktail of odors, other receptors will be activated, and you get a larger blimp. But still, it's only a fraction of the whole odor world around us, so only 1,000th of the receptors are expressed. In the transgenic mouse, we would expect zero response from the cocktail of odors, right? Assuming they don't activate the M71. If they do, maybe a little bit, but they don't. But if you give acetophenone, which is the ligand for this, it's going to explode, okay? So the mouse takes a sniff from acetophenone, the whole epithelium, all the axons, all the millions of axons start to fire like crazy. And indeed, you see a huge activation on the olfactory nerve, and this is highly quantitative, very clear, etc. So if you do live imaging on the glomeruli now, So you see that if you take a control mouse, these are these glomeruli, you emit from the dorsal surface. This is a crossed mouse that you can see the activation of the odors. And you give one odor, you'll see one glomerulus. You give a different odor, you see a different glomerulus. This is ethyl acetate, this is eugenol. And if you give isoamyl acetate, you get these two different glomeruli. And now you give the ligand, acetophenone, you get this glomerulus activated. And if you increase the concentration, you get a stronger activation. So this is a control mouse, okay? 4 different odors, 1, 2, 3, and 2 different concentrations of the ligand. And you see this combinatorial activation pattern, which we talked about. In the mouse that this monoclonal nose mouse, all other odors, at least these 3, did not induce any activation pattern. And when you give acetophenone, the whole glomeruli light up, okay? And then they did some control with by putting a GABA B receptor antagonist, so to silence the lateral inhibition. We'll talk about this later. And it just proves that the receptors are expressing and they're functional. Yes?
You said that we don't know for many of these receptors what are their functions.
What is their ligand?
What ligands are connected to?
Yes.
So my question is, why can't we theoretically create many of these like mouse, transgenic mouse with one receptor and to investigate these in that way? Like to see.
How are you going to do that?
To see the activity for different ligands when you're. The problem is. Every time you have one receptor and then you check the activity for different ligands.
Fantastic. But why don't do it in a petri dish with 96 well cells, like we told you we can do before? To express this gene in a cell is very easy. You run it through the microscope and you do 96 receptors every day. To make a mouse is probably, I don't know, $50,000 and takes 2 years to make one of these mice, a year and a half.
One of them or
one of them. Yeah, you can make a few in parallel, but then you need $2 million to make all these mice. So you need a good reason to make a mouse. And the reason that they made this mouse is to talk about whether there is a spatial code, a computational code in space. Because you will learn in the visual and auditory system of some beautiful maps, tonotopic maps and visuotopic maps, retinotopic maps, etc. And some people would claim that this is important for computations, okay? One concept, did you talk ever about lateral inhibition? Wow. Okay, basic stuff. So basically, you will learn this is a very basic thing in sensory systems that, for example, if neurons sit next to each other, then, for example, there is a clear tonotopy. So this neuron will be activated by low frequencies, let's say 5 kilohertz, this one by 7, this one by 9, 11, 13, 15, etc. You will learn the real values, but anyway, there is order here. And there are some processes of that is called lateral inhibition. There are inhibitory neurons in between. And this is one way to compute and create all kinds of nice computations. For example, things like lateral inhibition, where one input is activated. Okay, this would be the input would come here, and this would immediately inhibit the next door neighbors. Okay, so this will be inhibition here, and this is will enhance the contrast. So this is one very classical, basic physiological mechanism of contrast enhancement. For example, when you see an image between the edges of an image and things like that, and this is due to the perfect structure of the neurons in the brain and this lateral inhibition context. Here, and this you can see that, so for example, our Nature Neuroscience paper from Gideon, it shows exactly this. So the textbooks will tell you that there's a smooth gradient of neurons in the brain, which is in fact what you see in large scale, but if you look at fine scale, you see these neurons that are activated by different, completely different orders. So global structure and local disorder or chaos locally. And how is this important for computations in the brain, etc. is a whole other different level of discussion, but nevertheless, okay? But the issue here is that inputs are, like I showed you in the slides before, they come in a patchy manner. It's, again, a combinatorial code, okay? So you take, we talked about these, okay? You do imaging here, and when you give an order, so there is some kind of pattern which is activated in the brain. The question with the monoclonal mouse was whether this pattern is important or not, as it is, for example, in other cases. So what they do, they said, okay, we're going to wipe the order and make everything the same, everything homogeneous, expressing the same receptor. Great. When they tested these mice, they found deficits in olfactory perception, olfactory discrimination. These mice, so what they do here, this is a very easy and hard task in these three graphs. And what you can see is control in blue and the transgenic mice in red. And you can see that these mice can do well, perform well on very easy discrimination tasks for the mice. And when the task gets a little harder, they do there is a significant decrease. What? Discrimination of two different odors. In this case, it's 2% amyl acetate versus 1% pinene. This is, the top is 2% versus 1%, blah, 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 etc. It's just the concentrations of the odors. And if you start to make the difference smaller, then it becomes much harder. And what they do here is they do mixtures. Okay?
Why should they smell it at all? They don't smell it.
So they do. They are able to discriminate different odors. You saw this small blimp.
But they have only one.
That's the results. Okay, you do get some kind of blip in the small blue blimp, and they are able to do it. It's not a knockout. It's an overexpression of a single odorant receptor. Okay, and all the rest, there is very low levels of other receptors. But basically, what they wanted to test here is the importance. Is it important that the glomerulus is on the right side or on the left side? This is like a very canonical, basic thing in neuroscience. Is space important? And so this, of course, is not an elegant way to do that, because they don't just. The best thing would be to change the position of the players in the game. What they did here is a very massive manipulation, and made a mouse that expresses 95% of its neurons are expressing a single odorant receptor. It's also very sexy story about a mouse with a special detector for one component, etc. But nevertheless, they do manage to differentiate between different odors. Just when it's hard, they're having difficulties. One. The other amazing thing is that they cannot distinguish between acetophenone versus air. They cannot. They cannot distinguish between the ligand and air and anything, okay? So you see, let's say 0.5% acetophenone versus air, and a normal mouse can do it very easily. These mouse cannot do it. So the deficit is even worse in the receptor that they are expressing. So the claim of the authors is that the network is doing some kind of comparative computation. If everybody is doing the same, I cannot differentiate what is the odor and what is not. Okay? So in order to extract what it is, I need some kind of noise in the system, inject some kind of contrast and noise. And to take this point further, what they did is that they asked the mice to distinguish the same odors, let's say 0.5% acetophenone plus other odors, and then they can do it. And their claim here is that this patchy organization is important for downstream networks to read what is the information in the olfactory bulb. Yes?
Just another question about its monoclonal thing.
Yeah.
I'm excited about that. So,
It's great.
are we able basically today to create, let's say, dogs who smell only explosives or only certain kind of drugs or
Well,
in different ways?
first of all, you don't need to make them, to make to make them. And you even see that they perform worse than the regular wild type.
But not for the specific thing you want them to smell, as we saw before.
No, if you teach them to smell a specific odor, you can teach them to smell that odor, a normal dog.
But this way you don't need to teach them anything. Just whenever they smell, that's it.
Well, in fact, you see a deterioration in the performance. In fact, what you see is the opposite from what you say. You see a deterioration in the performance of these mice. They're not as good.
The graph we saw before.
It's just an activation. It's just the number of spikes.
You put an electrode in there.
No, you put an electrode, you see a blast of activation. But the brain cannot extract from that. A lot of activity doesn't mean it's very good.
We assume that the dog or we or whatever feels when there is a lot of activation. There is a
But that's exactly the point I'm trying to convey here. If you have 100 cells, you have 10 cells, and all of them are doing the same thing, okay? They will now fire 20 spikes. And I'm going to tell you what is the odor here. You can't tell what the odor here. The odor, it's important to distinguish an odor. It's important that this one will give 10 spikes, this one will give 7 spikes, this one will give 0, 0, 20, 15, and 1. Then you have this combination. You can extract what odor it is exactly. You give a different odor, the numbers change here. It's much richer. There's more variability. There is more information in this than in this mouse.
Then in a situation when all of the neurons over there are firing 20 against...
The mouse cannot say if it's an odor or not.
Yeah.
Okay? If you increase the concentration, it will be, it will increase from 5 to 10 to 20, etc. But everybody's doing the same, and there's less information. So more activity, of course, doesn't mean more information. That's for sure. Okay. Okay.
So can you explain again why the last flow is at the set of pinin plus something else is easier to discriminate?
No, what they say, it's exactly like these graphs. They can discriminate the other odors. It's just particularly the acetophenone that they have a hard time with. But when you combine it, if you inject a little bit in the context of the coding, if you inject a little bit, if you make this 22 now, then they will be able to do something.
So the little effect of other odors
Exactly. So you need the differential activation.
are more informative than
Yes.
combination.
Yes. Okay. So I'm reminding you, we're talking about olfaction. I talked a little bit about odors, a little bit epithelium with receptors, sending their axons to the olfactory bulb. And now let's move on to the next station. This is a nice image that my friend Leo took when we were in the lab. Basically what you see here is a slice of the olfactory bulb. 2 different neurons are stained here. This is a mouse made X money and so much time to express. Actually, you saw this mouse when we talked about how they discovered that one odorant receptor is expressed in one neuron. Okay, that was the evidence for this. Dun, 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 dun. Here, this one. Remember this one? They made a mouse with GFP expressed only in a single olfactory receptor. So now if you follow the axons of these cells from the epithelium, what you see is what I showed you there. Is a single glomerulus expressing GFP. Come on. Hello. Sorry. Next time we'll go faster. That's the GFP. These are all the axons. It's a bundle of axons converging into a single neuropile region. So these are all the green are presynaptic axons with their synapses. And the neurons that read this information are called mitral cells. And they are the projection neurons of the olfactory bulb. From these neurons, information then goes to the cortex. But what we now know about mitral cells is that the mitral cells themselves send a single dendrite, single apical dendrite, into a single glomerulus. Shoot.
What is the apical mean?
So, get some light here. Idan didn't talk about that, I guess. So a neuron, though some of these neurons are special, a classical neuron will have a soma, an axon, which is the presynaptic component normally, and you'll see in many cases, at least in cortex and cortical neurons, where a lot of computations are going on, two types of dendrites. These are called lateral dendrites, these, and this is called an apical dendrite, which normally it's like an apex. It goes, it's just by the morphological criteria. So these neurons too, they have basal dendrites and apical dendrites, but what is amazing about the system is that these are these glomeruli, okay? And they send only a single apical dendrite into a single glomerulus. So basically, it means that they are receiving input from a given receptor. Let's say this is the receptor number, I don't know, 745. The axons impinge here, and here it's 209, etc. There are 1,000 of these. Okay? And what I told you until now is odorant receptors send axons to a single glomerulus. You see this beautifully in this image. And if we label the mitral cells, you also see that they send a single dendrite into a single apical dendrite into a single glomerulus. Of course, each glomerulus will be innervated. The number has been updated. That's 9. 9 mitral cells will send a dendrite into a single glomerulus. So basically, there is huge convergence. With about 5,000 axons in the epithelium, all of them sending axons. They're so dense here, they're so packed that you cannot see a single axon. What you see here is bundles of axons, of thousands and thousands of axons, all converging onto a single glomerulus. And here about 10 neurons, or 9 neurons is the latest number. Per glomerulus, 9 neurons will send a single axon to read information from glomeruli, and this information will be carried out to cortex. Shoot.
The cilia that we talked about before, the receptors,
Uh-huh.
so computation-wise, they are only for amplification?
Yes, I think the structure, yeah, it's just to increase the surface area of the, to increase detection ability.
Not the structure, I mean, this convergence, so the whole system is like, so it's only for amplification?
Yeah, I think this is like a very basic, you want enough receptors throughout the epithelium to be able to detect a molecule that comes in there. Amplification is a word with meaning, so I don't want to say yes on something that I'm not sure. Okay, amplification is that there's really gain, and a molecule hits, and suddenly there's some processes that amplify this. But okay, anyway, yes.
So the 9 dendrites, they come from 9 different mitral
Yes, yes. So 9 different mitral cells. Okay, the number again I told you is they will project their dendrites into a single receptor, and a different 9 on average, of course, different 9 cells. I think one of the papers I gave you is about what they call coding of sister cells. What they call sister mitral cells are the same group of neurons that send their axons to the glomerulus, although I'm not sure I chose this paper. Anyway, this is a very nice histological example to show you the anatomy of the system. Okay, so what I told you so far, these are the olfactory receptors. If they express this specific receptor, they send their axons to a given glomerulus, and mitral cells read this information and sends it out to the olfactory tract and the cortex, to higher brain regions. This is a basic classical anatomical phenomena in the olfactory bulb. So mitral tufted cells send dendrites only into one. There are, of course, exceptions, but for the sake of this class, 1 is enough. Shoot. Glomerulus is what we call neuropile. It's a region without a cell body. It's where axons and dendrites meet, where synapses are formed. So it's basically an anatomical structure filled with only dendrites. There's no cell bodies. Cell bodies actually sit in the periphery here, Okay, this one's the cell body, and they send dendrites here, they send axons here, they send dendrites here. It's a meeting place of synapses. Okay, this is called. What's important here to realize, which starts our discussion about computations in this brain region, is that there are that although these, it seems like there is these now canonical information lines, okay, or columns of information, and this glomerulus is thought to be a processing unit because there's input of this order, and this guy reads information here and sends out to the cortex. So one may think that the mitral cells are only relay stations. They do nothing but pass on this information. However, this is the essence of neuroscience, okay? When you want to know what a circuit is doing, how the brain functions, you want to understand this input-output function. What is happening in this brain circuit, and what happens to the information that comes, in this case, from the nose and outside the olfactory bulb. And this is true. It's like a canonical thing that you want in any neural circuit that you want to dissect and to try to understand is what is the computation that is happening, that is occurring on the way. It can be many things, okay, depending on the circuit and the purpose of the circuit. But here, one very evident and clear thing is that, although I will show you some data about the history of this in the last 10 years, but basically one could think that these are just relay stations. Input, output devices, mitral cells simply collect the information from many olfactory receptors, and then now they are, more compact information will now send the information out. However, there is a lot of interneurons in the circuit. In fact, every circuit in the brain will have what we call projection neurons. This is like a very basic, your ABC kit for neuroscience. Okay, there's afferent input to the system. Inputs, afferent, A-F-F-E-R-E-N-T, afferent input. This is the information coming into the system. There is projection neurons. Normally, we call them projections because they project out of the circuit. And this is normally the output. And each and every circuit in the brain will also, or more, again, I don't want to say each and every, probably there is some small without, but most circuits in the brain will have interneurons, which are neurons that their whole structure, the dendrites and their axons, is within the circuit. They don't leave the circuit. The afferents come in from afar. The mitral cells go out to far distances, the projection neurons, and local interneurons are responsible for the local computations. And in fact, there is a massive network of inhibitory interneurons in the olfactory bulb. So these are shown here in red. Some of them will make contact with, between glomeruli, so maybe there is lateral interactions. Some of them can be close by. Some of them can be afar, distant glomeruli. Okay, so now different glomeruli will interact. Let's say there may be cells that connect the glomerulus of 745 to 209. Maybe, I don't know. Okay, they synapse here and here. They're local, etc., etc. And even more or worse than that, there is a huge number of centrifugal inputs. Centrifugal inputs is feedback into the system from higher brain areas, shown here in purple. Okay, and there's lots of neuromodulatory inputs coming to the system. Serotonin, acetylcholine, or adrenaline, etc. Okay?
So interneurons, are they usually inhibitory or not necessarily?
Usually, yes. Here we have also a small pool of excitatory interneurons. We'll talk about that soon in a few slides. Shoot.
Are interneurons as likely to be between two neighboring glomeruli than two distant ones?
Yes and no. There are some groups of interneurons which are local, which will be between two glomeruli. There are even some which if we have time, we will talk about them, which are responsible for computation within a single glomerulus. They don't even connect between. Some will connect neighboring glomeruli, and some interneurons will connect distant glomeruli. So you have the whole thing. I mean, c'est la vie. Life is complex, the brain is complex, and in every place that we go, the more you dig in, the more different subtypes of cells that we find, and each one is believed to do this computation and that computation. And if you want to understand a network, there are many things you have to punch in. Okay, we are at a point today in neuroscience where what Idan Segev talked about, which is more or less is bounded by a membrane, which is the single cell, we understand it pretty well. There is good theory, fairly good experimentation, and what Idan showed you is something which you can take home and say, I learned something new, because we really understand it fairly well. I'm not saying that there's nothing to further look into, but this is something we understand fairly well, starting from Hodgkin-Huxley, et cetera, et cetera. But once you go into the network, into the population of cells and how they code information, this is, I would say, the current challenges, or one of the biggest current challenges that we're facing. How to deal with this huge diversity and heterogeneity and complexity, which is not bounded by membranes.
So what are the two levels?
Okay. Oh, the two, what do you mean the two levels? Okay. Sorry, I didn't get to that. So now, if we can roughly divide the interneurons into, or they have been divided into two general groups, one at the input level, at the area of the glomeruli, which will send dendrites and axons into neighboring glomeruli or local glomeruli, etc. And this is considered to compute or do some kind of computation on the input arriving into the bomb. And there's a different level or a different group of inhibitory interneurons. Those are normally called granule cells, and they are making synapses on the basal dendrites. And they are considered to be modulating the output of the system. So there's input and there's output, and there's two levels of modulation on the input and on the output. Okay, one thing you can imagine is interneurons that make synapses on the axons themselves of the olfactory receptor neurons, and there are these synapses. Okay, so they make synapses on the ORNs, for example here. They make synapses on the mitral cells. They make synapses on their neighboring interneurons, and life is complicated. But this is the level of input. And the second level is at the level of output. One beautiful synapse from the cortex, for example, are cells that make synapses here on the axon hillock, okay, and where the spike is initiated. And so this is a classical thing of a synapse on the output of the cell. So there are different levels of modulation. Questions? You seem very unhappy. You seem very unhappy. No. Matan, yeah. I can help? Or this is a general mode? I see, okay. It's not me, it's you. Okay. Okay. So now let's see what kind of computations we think are happening here. So now we know the anatomy of the system. We know there's input, we know there's output, and also unique output. Okay, I told you single dendrite here with some lateral dendrites all over, and mitral cells that are going, sending out the information. One of the classical canonical models of computation was found by Kensako Mori. He's from Japan, from Tokyo, will be retiring, I think, in a year or two. Still very active, very good scientist. And what he did is made electrical recordings from mitral cells and challenged them. Again, I told you, it's a problem to face the whole chemical space. So what he did is he took a series of odors, which are different only by their carbon chain length. So you take, all of them are aldehydes. This is undecyl aldehyde with 11 carbons. This is decyl aldehyde with 10 carbons. Nona, octi, hepta, hexi, valle, buti, propa, etc. Okay, from 3 to 11 carbon chains. And the idea is that similar odors will activate similar regions in the brain, kind of like the tonotopy organization I talked about, okay, in the poster and here, that similar neurons with similar receptor fields will sit next to each other. If you remember the classical paper we talked about from Hubel and Wiesel, they stuck their electrode in the brain. They say many of the neurons sitting together, they have similar receptive field. So that's the basic idea here. And what they found in this paper, which is in fact a paper from 1995, I was exactly at your stage in 1995, and this paper came out showing that you can find the hotspot of the activation of the odors. For example, this neuron, this is a single neuron responding to 9 different odors. What you see here is the breathing cycle, which is a very canonical thing in olfaction. Cells are firing with the cycle of the sniff, okay, in bursts of action potentials. And if you see here, the odor is given here. So you see 2 seconds before, 5 seconds before, 2 seconds of odor, 5 seconds after the stimulus. Here, nothing much happened. Here I actually see inhibition, before and after, inhibition, strong inhibition, excitation, 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 inhibition, etc. Okay, you see this? Great. So the model was that there is a center of activation and a surround of inhibition. So the inhibition is strong next to the activation hotspot. Inhibition is strong, and it recedes slowly as you go away from the inhibition maximum. It's not what you see? What do you see? No, there's inhibition here and here, a little bit, strong, a little bit lower, and this would be the activation hotspot. Strong activation, semi-activation, strong inhibition, lower inhibition, etc. Not convinced, huh? Okay, I'm pretty much convinced. This is called center surround inhibition, if you will. Okay, so there is center on and a relatively strong inhibition on the sides. Okay, this is again in retina, in other brain regions. This is a classical canonical thing that you find. Excuse me? Like the on-off cells, exactly. Similar to the on-off cells. If you have an on-off cell and you shine in the middle, it will be activated. If you shine on the sides, you will get strong activation. And if you go away, the activation recedes. This is also called a computation which is called Mexican hat decorrelation. Okay? And you may have started looking at it in I don't know, the computational papers that or courses that you do. But the idea is that, I just want to touch upon that, and then I'll, just so you see where we're going. Okay, sorry. Let's say you have 2 receptive fields, this neuron and this neuron, and they have a common receptive field. Okay, the computation that is made here is what we call Mexican hat decorrelation. Loosen this up because it looks like a Mexican hat. Okay, and this is a way to decorrelate between similar inputs, where you basically amplify the center and you inhibit strongly the surround activity. There are other ways to decorrelate. You can do like a non-specific inhibition and decorrelate, dump the whole thing, and then still the overlap between the 2 stimuli will be smaller, but also the maximum amplitude of the different cells will be smaller. This is a different type of computation. And what we think, or at least the classical model of Mori, would argue, that there is this Mexican hat decorrelation, okay, which Matan doesn't see in the slides. All the rest do see it? Eventually you see it. Even you gave in. So this is the classical model. But now let's challenge the classical model. One paper, 1995, not long ago. Great. Let's make some assumptions. Okay. The classical view of decorrelation. The first prediction is that closely related odors, you remember the 3-carbon, 4-carbon, 5-carbon, etc., will activate neighboring glomeruli. That's the first approximation, okay? We want that, if this is true, this Mexican hat decorrelation, then this really has to be next to this, and this next to that, in order so we can build this smooth topographical information.
But even if we accept that, that depends on the criteria. As we said last lesson, some people may think it's carbon, number of carbons.
Yeah.
Think it's number of rings.
Yeah, okay, I just want to break what I told you, to tell you that it's not holding well. Yes, shoot.
I mean, I don't see the connection between the number of carbons and similarity of odors. Like if you're dealing with light, 2 photons is more than 1, and 3 is more than 2. But I don't understand why the 6 carbons in the chain means that it's more or less fragrant or whatever than 5.
Yeah, so I thought, it's a kosher question, valid question, and they're simply, perceptually, they smell alike. I mean, if I give you to smell a limonene, which is like a lemon, and nanoaldehyde, you'll say it's completely different. If I give you nonaldehyde and decaldehyde, they're, perceptually more similar than the limonene. So that's one thing, which may be semi-answering your question. And the other thing is that it seemed as a reasonable thing to do. Whatever the receptor, you see the receptor here, it's chemistry. There's a receptor, and the receptor has a ligand binding site, and the ligand has to fit in. So, the chances that this molecule and this molecule are more similar chemically made sense. With the perception and, seeing these results, this was like a dogma in the field. You go to a textbook, this is what you find.
But if you take behavior measurement, so if you'll give someone the same molecules here, let's say to discriminate between the 11 carbon.
That's still, it's in the realm of very, very easy.
Will it be easier to discriminate between the 11 and the 4, let's say, than the 11 and the
It's
9?
all extremely easy. It's going to be difficult for you to distinguish between this molecule at 70% and this molecule at 65%. That's hard.
But it's easy to discriminate
Very.
between all of them.
Yes,
Right.
very easy.
So we cannot say that according to our feeling anyway.
No, but if I give you a completely different, if I give you an alcohol to smell, tell you how different it is, so the distance between this and this will be smaller than the distance between this and a completely different molecule.
And I'll be able to say that.
Yeah, absolutely.
But not to say that the 10 carbon molecule is more similar to the 11 than
It's going to be very hard to tell that this
more. Yeah.
that this is the same distance. 9 and 10 and 10 and 11 are the same distance. This is exactly the thing that Noam Sobel did, which I talked in the first slide, and they found they did principal component analysis, and the principal component was not it was not the principal component that could explain the variation. The variability was not the carbon chain length. It's some bizarre chemical property of in chemistry. The angle of the eigenvector times the molecular weight and the heat, the temperature at which the molecule is boiling, something like that.
So the assumption that number of carbons is such an important
It's not unimportant, but this was the rationale of Mori when he did these experiments. Okay, so now let's try to challenge this. Oh my God, this is very sad. Ah la la. Okay, so let's say the question is whether closely related odors like the ones you saw before activate neighboring glomeruli. And at some point in the early 2000s, again, not too far away, there was this concept of chemotopy, like retinotopy and tonotopy, and they built a concept called chemotopy that odors of similar structure will be aligned in a similar manner, okay? And one of these experiments is this intrinsic signal imaging, where they took exactly what you were complaining about. C3 is 3 carbons, 4 carbons, 5 carbons, 6 carbons, 7 carbons, and this did this imaging on the olfactory bulb. And what they found is that there is some structure. So 3 carbons shown here in red are, the 4 is somewhere in between, the 5 is here, but 3 is also, there's a spot here and here, and 8 is here. So they made, there is some kind of cloud, like general organization. That was like the dogma in the field. Even in some papers today, you can find these chemotopy maps. Aldehydes are here, alcohols are here, ketones are here, but there's always this overlap which bugs you. So, it depends on the resolution that you look at the brain. At coarse resolution, maybe. The other examples came from looking at molecules which are pretty much exactly the same in structure, but that they are enantiomer. Okay, enantiomers are, they have exactly the exact structure. They just, one face. It's like looking on yourself in the mirror. One side of the molecule is different in the polarity or whatever. And when they did imaging on these molecules, they found, okay, plus minus fenchone. Okay, this is a plus, this is minus. This is carbon plus and minus, and this is heptanol plus and minus. They found if this is the map of activation of plus fenchone, this is the minus activation. And now there's a new glomerulus that activates here. So there's always overlap. Same glomerulus you will find similar activating glomeruli, but also other glomeruli. And these molecules are almost identical, completely identical. Okay? And in fact, this argues against a perfect chemotopy. Because now the new glomerulus that lights up is not just next to the shared one, but it's further away. It's a few millimeters away, or parts of millimeters away. So this started to show cracks in the activity. Now the model makes further predictions of Mori. And I think with that we'll take a break. Let's just talk about this first. Yeah. Okay, one prediction. What you see here is basically a diagrammatic prediction of 3 olfactory receptor neurons and how they are activated by the different carbon chain length molecules. So the prediction of Mori was that this olfactory receptor neuron will be activated much strongly by the 5 carbon chain molecule, a little bit less by the 4 and 6, a little bit less by 3 and 7, and by 8 it will be silent. Okay, and this is depicted here in this glomerulus, this ORN. Okay, this is number A, and this would be, so to speak, the responses of mitral cell A if there were no lateral interactions. There are no computations in the circuit. B is an olfactory receptor neuron with receptive field around the 7 carbon chain. C is the same thing with around 9 carbon chain. And the prediction is that if we record from the mitral cell that sends its dendrite into the B glomerulus, we will see this Mexican hat decorrelation. Okay, so you will get the input from B. That will be the center. But the surround molecules will inhibit it strongly. Everyone sees this? Okay. We're trying to understand this, or just if this makes sense, that's good. And we can take a 10 minutes break. Okay. Mexican Hadley correlation. Okay. So now let's try to challenge that. What these two groups did is took advantage. Now, instead of thinking, okay, maybe aldehydes of different carbon chain lengths are would make sense, let's do the real thing. Let's make flies in this case, because it's easy to make flies. Unlike a mouse, the life cycle, first of all, dogs you can't make transgenics. It will take forever. Yeah, you can make here and there, okay, but the life cycle, it takes a very long time for science. A pregnant mouse will be for 3 weeks, and you'll have a next generation. And flies is very fast. You can make, and you can actually make something like, I think don't get me wrong here. I mean, I'm recorded, so I may make maybe incorrect in the numbers. But in flies, you can put in something like 13, I think, different transgenes, different genetic combinations. In mice, it's very difficult. So you can do 2, 3, 4. 4 is starting to be a real headache. Dogs and things like that, I think the techniques are not very easy, common, cheap. It's much more complex. Okay, anyway, so they made these transgenic flies, which have one line of in the fly there are, you remember maybe or maybe not, there are 60 different olfactory receptors, not 1,000. So in terms of the number, it's an easier model to work with, to handle 60 than 1,000. And more than that, in mice, just in flies, you can identify each and every glomerulus by its specific subtypes by the morphology. So if I see this olfactory bulb, if I'm a fly neuroscientist, I can say that this is this is the glomerulus of odorant receptor number, 26, and this is 54, and you can identify it between different individual animals. What they made here is 2 different lines of flies, sorry. One with the dendrites of the mitral cells expressing a calcium indicator reporter, and one with the olfactory receptor neurons expressing the fluorescent indicator reporter. So they have input and they have the output of the cells. Okay, this is how one of these mice looks like. What? Yes, I can repeat it. So they make new flies now. You remember that the mice that Axel made were these monoclonal, everything expresses the same thing. Now they do like this. All of the mitral cells express the calcium indicator, okay? And all of the glomeruli will have this indicator for activity, each one with 9. This is one type of a fly. They make a second type of fly where all the axons have the indicator. And now they give an odor, and the odor will light up a given glomerulus, but they know exactly which one it is because it's a fly. It's very easy. It's 1 of 60, and they have a map. You have an atlas of the glomeruli, and you know that number 55 was lightened. This is number 56. This is 57, etc. So they can ask, how do activation patterns look like when you challenge the olfactory bulb, the system? The system is like that. You take one of these flies, you chop his head off, and you put the head of the mouse with the olfactory bulb on the top. You see the olfactory bulb is here, roughly. And this is the antenna of the mouse. This is the nostril of the fly, sorry. Every time I say mouse, it's fly in this slide. So this is the antenna where the olfactory receptors are, and they send their axons into the glomeruli. And they put this system under a microscope so they can image the glomeruli from above. And here they deliver different odors, okay, onto the nostrils or the antenna of the fly. And this is the type of data that they get. This is calcium imaging data. I can't find it. Or I can? No, I can't. Never mind. Okay. So when you give an order, this is a live image. You see this one flashing, this one flashing, this one flashing. Okay. It's pretty nice. Basically, so this is this is when the projection neurons. This is the fly. One line of flies with the projection neurons or the olfactory receptor neurons. Sorry, this is like the mitral cells, the projection neurons. They are labeled with the calcium dye, and this is when the sensory neurons are labeled with the calcium-activated dye. And they ask which glomeruli are activated when you activate the olfactory receptors and when you look at the activity of olfactory receptors and the mitral cells. And what they find here in this context, and this is a paper that came out in 2003 in Cell. Cell is perhaps the best journal in science. Okay, Cell and Nature and Science are like the top journals. I think Cell is even more in terms of the impact of it's considered very heavy and serious journal. Nature and Science, sometimes they have all kinds of sexy stories that are very interesting to the general audience, and they're put in. But Cell is normally, very heavy, 15 figures, and very long, and it's like classical papers. But in terms of the impact, it's very similar. So this came out. And the idea here was that basically the ORN to the projection neurons or mitral cell is a faithful transmission. So they are just simple relays. We can't see anything which is different between the input and the output. The exact same glomeruli get activated when we put one order, and the same mitral cells are activated when we put the same order. So their claim was that there is faithful transmission in the system. And this would imply that the system is a mere relay. There's no general computations going here. The issue was that the fly system is great in terms of genetics, but was a very difficult one in terms of recording electrophysiologically from the neurons. And this is calcium imaging. Calcium imaging takes just the tip of the iceberg and doesn't see the essence of the cells, which is the spiking activity and the electrical activity. And so this was the dogma, so to speak, of faithful transmission. So one can think, what is the role of all these interneurons in the system? Didn't make a lot of sense, but nevertheless, they said, and Richard Axel said, that's the system, that's the system. And then a big breakthrough came in. This is a girl at that time, young lady, your age, I think probably less than you for sure, going to work with Gilles Laurent. Gilles Laurent is a famous computational neuroscientist, at that time in Caltech. And she decides she's going to record from neurons in the fly with electrical activity, with microelectrodes. Everyone who tried before that failed. I think one of the only successful one was Axel Borst. I don't know if you were here in the seminar, recording from visual neurons in the fly, amazing system. But in Drosophila was a problem, because when you stick in the electrode, it's like, so the neurons sit in insects and invertebrates. Neurons sit, it's like a shkol of anavim, a grape, what? How do you say? Cluster of grapes or something. And they are surrounded by a small sheet of paper, of tissue. And when you put in an electrode, you need to break this thing. This is very tough. So you break this thing. And when you do, this whole thing goes out, and it's very ugly. Okay? So people couldn't really record from these cells. What they did is they played around with the cell line, the concentration of the tissue, etc., and could manage to open this and record for the first time from Drosophila. This was a big breakthrough, because until then, Drosophila was like the prep for genetics, whatever. You can put in these cells whatever you want in terms of genetics, but physiology was out of the scope. And Rachel Wilson, a young girl in Caltech, did her postdoc, and was the first one to be able to patch neurons in the fly. And this is a big breakthrough for the whole community, just technical advanced. But the question that she was asking is in olfaction, okay? And the first question was like, what is the input-output function of olfactory receptor neurons and their glomeruli? So she recorded, what she did is she now recorded from, she took a panel of 34 odors and recorded from olfactory sensory neurons and asked what is the width of the receptor fields of the olfactory receptor neurons. And what she found, in fact, is that, and she did this for the olfactory receptor neurons, shown in green, and she did this for the projection neurons, which are the mitral cells, the second-order neurons, which is shown here in red. And what she found is exactly the opposite of what she expected. Because you would expect, if the Mori model was true, okay, what she is doing now is asking what is the receptor field of ORNs, of sensory neurons, and what is the receptor fields of projection neurons. And you would expect that the receptor field of the mitral cell will be a little bit thinner because of this lateral inhibition, right? So she said, if the Mori model is true, if this concept is true, then I would expect thinner shoulders on the receptor fields of the mitral cells. And what she finds is exactly the opposite. Is that so now what you see here is a given cell, and she puts the best responding odor in the middle, and just for the sake of the clarity of observation, she puts the next ones on the shoulder, the next ones on the shoulder, the next ones again, so you get like a sort of a receptor field, okay? And this is the number of the stimulus that activated this cell, the number of spikes. So you see if here it's the first few milliseconds, I know, the first 230, and then you go more and more time, you get a wider range. But nevertheless, the idea here is that always the projection neurons had wider receptor fields than the sensory neurons, which is exactly the opposite of what one would expect from the Mori model. This was a science paper. Again, it was a technical breakthrough because the ability to record from flies, but also conceptually it was a challenge, if you will, to the concept of the Mori model. She then went further and did double recordings from the olfactory receptor neuron, sending the axons to the glomerulus, and the mitral cell, or the projection neuron, that sends its dendrites from the glomerulus. So basically, she's recording from here and from here, and she has the input-output function of the cell. Okay? And she finds that the input-output function of the cell is not a mere relay. It's not a linear relationship between these two, but this kind of relationship, where the dynamic range of the ORNs, not only do they respond to less odors, but even for a given odor, if she increases, let's say, the concentration, or how many spikes she puts inside an ORN versus how much spikes she reads in the mitral cell, she sees this kind of response. That the ORNs uses a rather narrow dynamic range, while the projection neurons, the mitral cells, use a much more wide dynamic range. Okay, and all the computations from now on, if you look at Rachel Wilson's paper, will be working on this kind of input-output function. Okay, she now moved from, I think she's probably much younger than I am. She is now in Harvard, a professor in Harvard, and has a very fruitful lab. So she's really a leader in the field, both of olfaction and also in putting in computational neuroscience into olfaction. She's very much like, I would say, like you, like the ICNC type of a student, which uses computations, but here also experiments. Okay. The issue is that when this is challenged in mice, we still don't get the same picture. So in mice, this has been done only once in this neuron paper from 2010. You see we're starting to scrape these days now. And now this is a work from Min Lu from China. Challenge the mitral cells with 100 odorants and set them exactly to the same in the same manner that you saw with the flies, and they see that mitral cells are very narrowly tuned, so they are very sparse. They will be activated only by a very few odors. This is mouse versus fly, which you saw before. No, in that slide. This is a mouse now, and you see the proof on the left-hand side. When he does this to the olfactory receptor neurons, this is a much harder experiment because he takes these receptors from this mouse that I showed you before, and he puts the cells in a petri dish and has to patch the petri dish, so it's harder. These are just randomly patched mitral cells. So the question, it's still out there. Is it faithful transmission, or is it not faithful transmission? Again, I mean, it depends a little bit on odor concentration, but let's not go on there. What? Repeat what you see in the graph. Yes, on the x-axis is the number of odorant out of 100, which he tested. Okay, he tested 100. On the y-axis is the number of spikes, the amplitude of the response with electrophysiology from a single mitral cell. And as you can see, a mitral cell responds with a very narrowly tuned receptive field. Not she. This is he. This is Min Lu. It's a Chinese guy now.
He didn't check the response of ORN?
Yes. So he also checked the response of ORNs, but it's not an odorant. There are 100 different odorants here. Oh yeah. This is the odorant for this glomerulus. This is the M71, the green glomerulus. He's putting his electrode next to this glomerulus. Let's go back to the image. Sorry. One second. This is the experiment he's doing. This is the mouse, for the sake of this argument. And he puts his electrode around this glomerulus here, trying to hit the ligand of M71. Okay, that's the graphs you saw. But also he takes these cells into the petri dish and tries to look what order they're activated, and he finds, as expected, that they're activated by the ligand. Okay, now let's go forward. We're back to the graph. Just one sec. Okay, so this particular odorant is 7-octanol, and he sees strong response when he looks at the olfactory receptor neurons, very little, if any, response to other odors. And when he's doing it for the mitral cells, now it's easy. He can check 100 odorants, not, 6. He sees also that they are responding to this ligand, which is where the arrow is. So what he claims is that he does not see this widening of the receptive fields of the mitral cells that one sees in the fly. Okay? Questions?
You can't record from the inner neuron?
This is what we do in the lab. Yeah. Okay. Other predictions. Marcus Meister from Harvard says, okay, this is what you say, classical MOE model, there should be an on-off classical retina thing, what we call a center, excitatory center inhibitory surround to these cells. And if so, if this is what you suggest, if this is the model you're going to teach students in ICNC, then we should get, on average, more inhibition than we get excitation. On average, because we really don't understand and don't know which odor activates where. Let's take a whole lot of odors, challenge many mitral cells, and you will get, on average, more inhibition than excitation. And in fact, when you do that, you don't get more excitation than inhibition. You get very sparse inhibition and very sparse excitation.
But even if you have on and off,
Yeah.
on center and off periphery, how do you know that it looks like this? I mean,
No, that's the dogma. The dogma of the MOE model.
why wouldn't the red
The?
Circles and BB.
Yeah. But we're talking about glomeruli. And we're talking that a single glomerulus is activated by a given odor, like that. And the surrounding glomeruli around him, this will be one glomerulus, and here will be a few more glomeruli. Okay? And the concept was this. And he said, okay, if that's true, then I expect to see more inhibition than excitation. And in fact, what you see is the same number of excitation and inhibition. What you see here is 25 different odorants recording from a single neuron. And this is the time, 5 seconds before odor onset, 2 seconds or 3 seconds odor onset, and this is color-coded for the spike rate. And you can see that this particular neuron responded with excitation to 3 odors, one with strong excitation, one with little bit, one with medium level. And on the top, there is 2 inhibitory. I mean, the colors are not very strong, but there is 2 inhibitory signals as well. So the claim here is that there is relatively similar levels of excitation and inhibition, and at least this suggests that there is no massive lateral inhibition like in the center surround model. Furthermore, this is true for awake animals. I'll skip that. Furthermore, what he did now, Marcus Meister in Harvard, is patched a neuron, a mitral cell in the center here. He said, okay, this is the model I want to test or refute. Let's patch that, and let's now do imaging, not only electrophysiology, but imaging to see exactly which glomerulus is activated. And I'm going to assume that I'm going to have a center excitatory. One odor or a few odors are going to be excitatory. But if I'm going to hit, by chance, this glomerulus, I'm going to get inhibition, like Maury suggested. And in fact, when he does that, this is the image that he gets. So he does see a very strong center excitatory, a very strong excitatory center. But he sees a very sparse and patchy surround inhibition, which is not center surround, but it has this different topology. Some of these can be, a very strong excitatory center, maybe another excitatory region, but always a very single, strong excitatory center. But inhibition can come from different places in the olfactory bulb. Yeah.
How did he find the inhibition?
So he records from the mitral cell, and he flashes. He activates. Now you can do this with optogenetics, where you can really flash and excite different glomeruli. And you see if you get excitation or inhibition in the mitral cell. You just count the spikes.
For the light one, he got inhibition.
Inhibition, yes. For when he activated this glomerulus, he got inhibition in this mitral cell. Yeah.
So basically, what does this mean? I mean, besides the fact that we see that there's less glomeruli, this inhibition that are connected comparing B and C, this geometrical, what does it mean?
This is for you to say. Okay? We are now trying to explore a new neural system.
Okay. So no,
That's going to be your job next year. And you're going to come to a circuit where you know maybe the anatomy, where you have an electrode where you can record, and you're going to have to try to figure out what's happening here. I'm telling you a story on the olfactory system. At this moment in time, there are 3 dogmas in the field. There are scientists which will tell you that there is center surround inhibition. This, I think, more and more people will now say that it's certainly not, it doesn't hold at single glomerular resolution. Another group of people which will tell you that there is inhibition, but it's not topographically organized. There are no strict rules when you look locally in the circuit. In fact, this I may not record this. Okay. In fact, I am from this region. And this is something we try to convey here, and we are fighting very big people in the field who feed the whole network and the whole community that there is beautiful organization in the brain, and the 6 kilohertz neuron sits next to a 5.9 kilohertz neuron duplicate, etc., etc. What we say is that this is redundant information, computationally speaking. And in fact, the system is much more heterogeneous, and this heterogeneity is not noise that one should cancel out. People see these cells, let's put it out because it interferes with our computations. The techniques today are so strong that you can really take a circuit and analyze it at single cell resolution, and what we see is different things than what I was born on. Okay? Because when I was in your place, this is what they told me. There's a perfect tonotopy, and this is the computation. The inhibitor neuron sends it in right here and here, and there's lateral inhibition, and a Mexican hand decorrelation, blah, blah, blah. But I would say that my role to you is to teach you or show you, to challenge the dogma, to tell me why am I wrong, or why the thing that you are going to start studying in your master's or PhD, you have to challenge it. And don't take things as for granted. That's the whole idea, okay? And I'm also, because you can go to the web and learn about olfactory systems, which is, in many cases what you're going to do, because I'm telling you some of the basic stuff. But more importantly, at this stage of your career, is to take what I'm saying and tear it apart and throw it to the garbage. If you do that, that's the mission. Besides the exam, where you have to answer the question correctly. The third dogma, I'm going to get to it, is local circuits. So the third dogma is that each and every glomerulus has his own local computation system to decorrelate the signal. Okay, the basic idea is that the system, if two similar odors come in, they will activate similar patterns of activation, but the mission of the animal, the perceptual mission, so to speak, is to differentiate between these two very similar patterns of input. So the idea is that we need to decorrelate. It can be a Mexican hand decorrelation, it can be otherwise, but the idea is to decorrelate. So one way to decorrelate it is this computation. Another way, which we still don't clearly understand, like Adi said, where are you taking me? Hello, this I can understand, but don't sell me this. I'm just selling you this because this is what we see in experiments, okay? The other theoretical one is that each glomerulus has its own local computation, which I will get to in a few slides. That's like the three different flavors of computations going around. Okay, so this, The other thing is anatomical, about the topography. I mean, by now you already see that there are big holes in the concept. The other thing was that, only nearest neighbors will do the inhibition, and, I'm going on and on. Now, one can do a new method, which is called transsynaptic tracing, which one can label a cell, which we call a starter cell. And then after a while, this virus can jump to the presynapses and label the presynapses. And basically, one gets the anatomy of the synaptic connectivity of a given neuron. This is one of the first options, techniques with this rabies virus. It's based on a rabies virus, Kalevet. I myself have 3 shots because I started to work with it. If you're going to come to my lab to work with me, you're going to get some shots, okay, if you want to do tracing, anatomical tracing. Anyway, what you can do is you can trace the synaptic connectivity from the cells, and what one would assume if there is the center surround is that if you label this cell, you're going to get the cells, synapsing onto this cell are going to be the ones close by. But what they found is this type of, which works well with the physiology I showed you in the last slide, okay? So the cell gets inhibitory inputs from many regions which are far away. In fact, they're in these bunches of neurons, and they say it's similar to the columns in the cortex, but anyway, let's just think about that. That's the concept. Okay, another prediction. Lateral interactions are strictly inhibitory. This, of course, you can clearly see from Rachel Wilson's fly recordings that that's not true, because there is expansion of the receptive field. And in fact, the fly people, now I'm back to the fly, okay? We're bouncing back between fly and mouse. Fly, again, cell paper. This is a Nature paper, I think, Olsen et al. What they did is the following. They made a new fly. Instead of having all the receptors expressing a given receptor, they wiped out all of the receptors but one. So all of the olfactory receptors now have a non-functional odorant receptor. They cannot smell anything, anosmic, besides one olfactory receptor expressing one odorant receptor. And what they did is they now patch this cell. Of course, they get the ligand that activates this, no problem. But what happens if they patch a next door mitral cell? What would happen? If there is inhibition, lateral inhibition, what? If I'm recording from this mitral cell, this is shut down. This is dead. There's nothing here. The only activity is here. You get inhibition, okay? If there is lateral inhibition, you get inhibition, because this cell now gets activated and it inhibits it, so they predicted inhibition. And of course, if there is lateral excitation, you will get excitation. And what they found is that they get excitation. So when you record from here, you get that excitation from here. But when you record from here, you also get excitation. And this is the basis of the widening of the receptive field. And another group, this came out in the same year because they did these flies, while these guys found the exact cells which exert excitation, because they found an excitatory neurotransmission of local interneurons. So for one of you who asked whether local interneurons are always inhibitory, the answer is clearly no. Okay? So they found excitatory interneurons in the fly. Right.
What's the difference between this technique and the monoclonal we saw before?
The monoclonal was making all of the receptors the same, express this purple receptor. Now they wiped out the whole thing and left only intact the regular normal receptor in place, in order not to make a clean experimental system, not to contaminate it, so to speak. Yeah.
What function does excitatory lateral excitation serve?
That's a great question. There are some computations that she talks about, but that's not, the jury is still out on that. Okay? But that for sure explains the phenomena of the widening of the receptive fields. It's a little bit more complicated than that because there is an aspect of time which kicks in. But this is, I think, for more advanced topics. In mouse, now we're back to in mammals. In mammals, they could not find, or we still could not, nobody could find lateral excitation. In the mouse, at least, in the bulb of the mouse, all of the data that we have so far is lateral inhibition. One example of a very nice paper is double patching of two mitral cells that go to different glomeruli. This is done in the slice, not in vivo. Okay, you patch this neuron that sends this dendrite to this glomerulus, and this neuron that sends this glomerulus, and you give spikes. You place spikes here, and you never see excitatory responses in the next door neighbor. While if you happen to patch same sister mitral cells that send excitation to the same glomerulus, and then you find, they call it excitatory spillover or something like that. Okay, so this, at least until now, nobody could find excitatory lateral interneurons in the mouse. The main idea is now, so I'm summing up what I told you, different olfactory receptors are intermingled in the olfactory epithelium. All of the ones that express the same receptor will send axons to converge onto the same glomerulus, shown here with the different colors. From here, mitral cells will send a single apical dendrite to the glomerulus, many lateral dendrites, and there's a whole lot of computations going on here. In mammals, we see sparse receptor fields from mitral cells. In flies, we see broad receptor fields from mitral cells. Why is this working in this scenario and that in that scenario? We still don't know. In the mouse, there is quite a bit of work on interneurons. I thought that if we had time, and we don't have time, I can tell you about one type of interneurons that we're studying today. In my lab, these parvalbumin positive interneurons shown here, just between the input layer and the output layer, which exert in a very interesting phenomena, a very widely distributed inhibition throughout the bulb. Okay, so let's see. Let's look at. Let's jump here because we talked about that. Two different scenarios. This is the dominating scenario in the field, but many things would refute or challenge the classical dogma, certainly via the center-surround inhibition. Yeah. The other one is the last option that I talked about, is this local glomerulus, looking at the glomerulus as a single functional unit. And basically, they claim that this will achieve and these guys are theoreticians, God forbid, doing only theory. How did Chaim Sompolinsky tell me? If you want to do good theory, avoid the data at all costs. It just interferes with the theory. Okay. So this is now at the theoretical level. There's some evidence for it, but no solid evidence. Okay, so the idea here is that glomeruli so this is the Mr. glomerulus. Okay, this is the olfactory receptor neuron sending its axon in here. Many of these, right? This is a glomerulus now. And here there is a mitral cell, dendrite. And the theory by Tom Cleland, which is a very smart guy, wrote a very nice paper like here, is that the periglomerular neurons, they are very small cells, and they get input from the olfactory receptor neurons, but they themselves exert output inhibition onto the mitral cells. Now they are small, compact, with high input resistance, and therefore will be highly activated by the odor activity. Okay? So a weak odor activity will cause them to fire way before the mitral cells, because they fire and the mitral cells are inhibited by them. Okay, this is shown here in the weak potency or even moderate potency. If an odor activates a glomerulus in a moderate way, you actually see net inhibition on the mitral cell, because this activity excites the inhibitory neuron, which then inhibits the mitral cell. Now, the ORNs, the olfactory receptor neurons, also excite the mitral cells, and only strong activation of the olfactory receptor neurons, which kicks in at strong potency or strong odorant excitation, will excite this synapse harder, and the PGNs reach some kind of plateau, and they can no longer override